All right, welcome to chapter 11, where we're going to talk about the cardiovascular system. This chapter is a pretty fun chapter. Most people at least enjoy the cardiovascular system chapter because this one in particular, it says cardiovascular system, we'll talk about the heart and blood vessels, but realize there's a lot more. As with some of these chapters, there's a lot more to learn. But I'm going to talk a little bit about a few things that are not in your book or may not be covered really well in your book or just kind of is glossed over. Just because I've found that students have, always request a little bit more information about it or ask a few questions there. So I'm going to talk a little bit about a couple of extra things in here, but don't sweat it. It's not going to be something that is going to be front and center on a test or anything like that. And if there is something that I ask about on a test that does relate back to this material, by me talking about it in this lecture, you'll understand it a whole lot better and you'll probably do better on that particular item or two that may be there. So, all right, so let's get started. Obviously, we're going to talk about the cardiovascular system in general as a whole. You need to realize that it's a closed system. Has the heart and the blood vessels. Last chapter we talked about the blood that's in the vessels. This chapter we're going to talk about the system that carries it around or moves it around the body. The heart's the pump. Hopefully that is not a huge surprise to anybody. And of course the blood vessels actually allow the blood to circulate to all parts of the body, throughout the body. The cardiovascular system functions to deliver oxygen and nutrients to the cells and the tissues. It also removes carbon dioxide and some other waste products from the cells. Remember we talked about blood, that connective tissue that's called blood. Does that, but the cardiovascular system plays a role in actually moving the blood around, making it possible for the blood to do that. So both of them serve that same role. All right, heart is located between the lungs in the inferior mediastinum. Of course, it's in the thorax, it's in your chest. It sets a little bit, just a smidgen to the left rather than dead center. It also is partly responsible for why the right lung has 3 lobes and the left lung only has 2. And the right lung is a smidgen bigger than the left is because the heart sets a little bit to the left and takes up a little bit of that space. The apex of the heart, the pointy part, the far end, the distal end of the heart, actually points more towards the left hip. So our heart doesn't set straight up and down in there. It sets on an axis. It's kind of tilted. And of course the base points towards the right shoulder. So when we look at the heart, when we want to get a good look at the heart, it goes from the shoulder to the hip. And I'll talk about that being kind of important later when we put somebody on a heart monitor, to look at the squiggly lines that their heart makes. And the way that we put somebody on a heart monitor matters. Okay? It's roughly the size of a human fist. Of course, there are some conditions that can affect its size that, it's going to vary from person to person. Here's a pretty good picture of it, and you can kind of see how it sets a little bit to the left. The pointed part down there at the bottom is the apex of it, the apex of the heart. You'll hear that referred to, not all the time, but from time to time you'll hear the apex of the heart referred to, but it points down this way towards the left hip, and the base is kind of the left hip and the base is towards the right shoulder. So we see kind of this angle, this tilted axis is the way that the heart sets, because when we divide the chambers, the heart's kind of divided like this, a little bit more so, and that's pretty crude, but you kind of get the idea of the way that the heart's setting in there at a little bit of an angle. It's not perfectly straight. And there's a view of it a little bit further back. Okay, so the heart is surrounded by a pericardium. It's a double-walled sac, pretty fibrous, but it is loose and superficial. And there's a membrane that's a little bit deeper that's called the pericardium. It actually surrounds the heart. The fibrous, there's a membrane that's further in deeper inside of the pericardium that's divided into a couple layers. We have the parietal pericardium, which actually is on the outside layer. It's on the pericardium itself. This sac that's covering the heart it's kind of a thick, kind of leathery sac, similar to the meninges of the brain. It's just a protective sac that allows the heart actually to beat inside of there and move inside of this sac to where that it's not rubbing up against the lungs every time that it beats, because a heartbeat is a fairly violent thing, so the heart moves around inside of there, at least moves into a small degree. Without this sac, there would be a lot of friction going on in there, so this sac actually provides a place for the heart to beat while still protecting organs around it and protecting the heart itself. So inside of there, we have a membrane that covers the pericardium side and the heart side, the visceral pericardium that's next to the heart. It's just those two membranes that separate the sac from the heart. We see that similarly. We had very similar in the brain when we studied the brain. And then there's some fluid, some serous fluid, that fills that space in between those layers that provides the friction. It's a really slippery fluid. It's similar to blood plasma, and it really kind of is blood plasma, only it's super filtered and those types of things. But that fluid can actually get an infection, and we can have an infection in that fluid, in that space around the heart or even around the lungs. So it's possible for there to be problems there. So a little closer look at those membranes and how that covers the heart. The interesting thing about this membrane is the membrane doesn't actually completely surround the heart. It's kind of like if you took a balloon and you pushed your hand into the balloon, and the lining of the balloon surrounded your heart. The top part of your hand up around your wrist actually it wouldn't be surrounded by the balloon, because that's the entry point. Well, that's kind of what happens up here at the top where these great vessels are, right up in here. The pericardium actually doesn't surround those vessels. So it's almost like the heart is setting in this little sac, like it's been placed in a bag or something like that, and the top is kind of open, so to speak. It's not really open because the doggone thing's attached. The pericardium is actually attached to the heart, so there's not really an opening. It's kind of a virtual opening, but it attaches to the vessels right there, kind of surround it. Another thing I want to point out is you see all this down here, this connective tissue down here. Just so you're aware, the heart, the lungs, and all those different things are held in place by tendons and ligaments and that kind of stuff, primarily ligaments that hold them in place. This big vessel right here, there's a ligament that kind of surrounds this vessel a little bit, holds it in place. That way all of these organs stay where they're supposed to be. It's not like they just magically know where to stay. They actually have something holding them in place. Just like the joints in our body, if we didn't have tendons and ligaments holding our joints in place, they wouldn't stay in place. There's a lot of athletes that have not had their joints stay where they're supposed to be when they've damaged those things, and they've had to go get them fixed, and maybe some of you guys have too. So some of our organs in our body, even our liver and our spleen and our intestines and all that stuff, are held in place by this fibrous mesh, so that they don't go moving around where they don't belong, because that would be a big problem. So 3 layers of the heart wall. When we look at the heart wall itself, it can be divided into 3 layers. We have the epicardium, which is the outside layer. And then we have the myocardium, which is the middle layer, and then the endocardium, which is the inside layer, also known as the endothelium. So these are just layers of the heart. Of course, the myocardium is the middle layer, and it's nearly entirely cardiac muscle, whereas the epicardium is kind of a connective tissue layer, membranous kind of thing, and so is the endocardium, kind of more of a membrane than anything. So where all the muscle is at is the myocardium. Of course, myo means muscle, so that's where all the muscle is. The heart also is divided into 4 chambers. And the left and right side of the heart actually act as 2 separate pumps. It's a 4-chambered heart, but each side acts relatively independent of the other, but they really kind of don't, because of the way the electrical system or the nervous system kind of makes things work. So what we have is on the top of the heart, the top 2 small chambers, there's an atria on the right and left side of the heart, and we call those the receiving chambers. I also like to call them the priming chambers too, and I'll explain that here in a minute. And then we have the ventricles, which are the discharging chambers, which send the blood out to the body. So the atria on the top of each of the ventricles, there's an atria that sits on top of the right and left ventricle. And when we're talking about right and left, we're talking about the patient's right and left, not yours. Whenever we're referencing right and left on a person, we're talking about their right and left, not your right and left, okay? So we have an atria on top of each of the right and left ventricles. And that atria that's on top of that ventricle actually feeds that ventricle, so to speak. The job of the atria is to receive blood and act as a small holding chamber, very small, because when the valves open, when the ventricle squeezes the blood out and then goes back to its normal shape, it actually sucks blood in kind of like a suction. And then the atria squeeze, the little chambers on top of the ventricles, they squeeze and kind of finish filling up the ventricles. The ventricles kind of roughly fill up about 70% on their own just by the suction. Just by when they open up, they fill up about 70% on their own. The atria tops off that last 30% of blood that they can hold, so it kind of finishes filling them up. So that's really the job that the atria plays. It receives the blood and it actually tops off the ventricles. It fills them up. So, let's see. And here's just kind of an example, just a little bit of that. Okay. So this is actually a pretty good view here if I get my pen going here. This is the left atria. I'm sorry, it's the right atria, and this is the left atria. This one's the right, this one's the left. And we can look at those. They're roughly about the same size. Look here, these things are not real thick. There's not a lot of muscle to them, not a lot of musculature. Because their only job is to take the blood that's in here and pump it into here. That's their only job. This blood that's in here gets pumped into here, and they do it at the same time. So the right and left atria are going to squeeze at the same time, and they're going to push their blood into the ventricles, the right and left ventricles. Of course, we can see that this left ventricle is a lot thicker than the right. And the reason that is this left one is actually pumping blood to, left one's pumping blood to the heart, to the lungs, and the right one's pumping blood. Wow, I've got my right and left all backwards standing here looking at this, and you'd think I'd get it right. The right one's actually pumping blood to the lungs, so it doesn't have very far to pump blood, so there's not a lot of muscle that's needed to do that. The left ventricle, on the other hand, is pumping blood throughout the rest of the entire body, all the way down to your pinky toe. So it requires a whole lot more musculature to get the pressure that's needed to actually get blood moving all the way down there in your feet. And that's especially true when you are laying down or that kind of thing, or if you're standing up, the gravity and all that stuff has an effect on the blood in your arteries and in your veins. So that's what your atria and ventricle are doing, and that's kind of the makeup of those. And you can actually see these two valves right here. This valve is the valve that gives probably the most trouble, the mitral valve. This valve is notorious for these tendons, these chordinae tendineae, a neat word to try to remember. Chordinae tendineae, it's the attachment points of this valve. These sometimes will get stretched out, or they'll tear, or something will happen to this valve here. And this valve can actually do something that's called prolapse, or it can begin to leak, that kind of thing. So when the ventricle actually squeezes, when it squeezes in and it's trying to push the blood out this way, it actually pushes the blood back up into the atria instead of out of the heart. Kind of a crazy deal. It actually pushes it, it pushes it out this way and not this way. It's coming in this way. So the blood actually is supposed to go out this way, but it goes backwards because that valve is damaged right there. So mitral valve prolapse or mitral valve issues, those kinds of things. Oftentimes these mitral valves are also associated, usually there's some sort of coronary blockage. There's a blockage in one of these vessels that's close to that. One of the indications sometimes, not always, sometimes is atrial fib. Sometimes we see atrial fib and a mitral valve problem go together. Now that doesn't mean that because somebody has atrial fib, they have a mitral valve problem. That's not true. And it doesn't mean that if somebody has a mitral valve problem, they'll have atrial fib. It's just that those two are sometimes seen together. For whatever reason, sometimes they go hand in hand, but it's not a guarantee. So we can see the musculature. We do a cross section of the heart. You can see that left ventricle has a lot more muscle than the right. Remember, the left is pumping to the whole body. The right is only pumping to the lungs. So it doesn't have as much work to do. No less important, but not as much work. So then we have that division between the two ventricles. We call that the intraventricular septum. And then the septum between the atria is the atrial septum. Most of the time we just call this the septum, is often how it's referred to. And usually, though there's a thick musculature in there, most of the time it's the left ventricle that's using most of that musculature and not so much the right ventricle. But with the atria, they kind of share that. Remember that the atria are going to contract at the same time and the ventricles are going to contract at the same time. That's just the way they work. Okay? And then we've got this circulation path called the pulmonary circulation. When blood comes out of the right side of the heart, it goes to the lungs and then it comes back to the left side of the heart. So blood comes back from the body, it comes in the vena cava, and it comes to the right side of the heart. The blood that's returning from everywhere in your body, it comes to the right side of your heart, into the right atria, into the right ventricle. When it's pumped out of the right ventricle, it goes to the lungs. Now what happens in the lungs is all of that carbon dioxide that it's picked up throughout the body from cellular metabolism and everything that's going on, that CO2 that it's picked up, it's going to offload all that CO2 in the lungs and take on more oxygen. Part of the gas exchange process that's going on in there. Well, and we'll talk more about the gas exchange process when we get to the lungs. But just realize that we're getting rid of some CO2, or we're getting rid of probably a lot of CO2, and we're taking on a lot of oxygen. So the blood that's returning back to the heart, back to the left side of the heart, the left atria, is really rich in blood. It's got a lot of blood. So then that blood goes in the left atria into the left ventricle, and then it's pumped out of the heart into the systemic tissues, out to the rest of the body, and then that's going to return to the right atrium via those veins in the body. So blood is pumped out through arteries. Any blood going away from the heart is an artery. Any blood coming back to the heart is a vein. Sounds kind of weird. Don't always think of blood, arteries and veins. Arteries don't always carry oxygen-rich blood, and veins don't always carry blood rich in CO2. Because the blood that's actually going from the heart to the lungs doesn't have a lot of oxygen and it has a lot of CO2. And the blood coming back to the heart in the pulmonary veins has a lot of oxygen in it. So just remember it as the arteries, or the vessels that are going away from the heart are arteries. The vessels coming back to the heart are veins. So in the pulmonary circulation, there's a pulmonary artery that goes to the lungs, picks up more oxygen, and the vein returns it back to the heart. It's a short trip, it's a short path, and that's why the left side of the heart doesn't have to be so large. And we can see that here. Of course, this isn't a really great picture just because it depicts the systemic circulation and the pulmonary circulation roughly the same size, but really they're not. They're not even close. The pulmonary circuit's it's small, speaking. There's a lot of vessels in the lungs. Please do realize that there is a lot of vessels, a lot of capillaries, a lot of things going on there in the lungs. However, it's not near as large or spanning such a wide space as the systemic circulation does. So this shows us oxygen rich and oxygen poor, but remember, arteries go away from the heart, veins return to the heart. Okay? We do have some valves. I kind of pointed out a couple of valves. The thing that I want you to realize about valves, anytime you hear valves, Valves in the human body have to do with keeping fluid from going the wrong direction. Wherever they're at, they keep fluid from going the wrong direction. That's what valves do. That is extremely true in the heart. There's 4 of them. There's the atrioventricular valves. It's the valves between the atria and the ventricles. 2 types of those. Bicuspid, which is the mitral valve. We talked about that one on the left side of the heart. The other one is a tricuspid valve. It's on the right side between the right atria and the right ventricle. Now, between the ventricles and the arteries that are going out of the heart or away from the heart, those are semilunar valves. And then we have a pulmonary semilunar valve, which is kind of easy to remember. That comes off the right ventricle. Then we have the aortic semilunar valve, which comes off the left because it goes into the aorta. So when you're trying to keep these straight, remember the mitral valve or the bicuspid valve separates the left side, left atria and ventricle. Tricuspid is the right. Pulmonary semilunar valve goes to the pulmonary side, and the aortic semilunar valve goes to the systemic side. Hopefully that helps. But remember, valves are designed to keep things from going the wrong way. Those atrioventricular valves, the ones between the atria and the ventricles, are held in place by those chordinae tendineae, or heartstrings. So when you hear somebody talking about, oh, they really pulled at my heartstrings, that is actually something that does exist, and it's the anchors that hold those valves in place or keep them from going backwards, from inverting backwards. Those are open while the heart is relaxed, and they close during ventricular contractions. Actually, the force of the ventricles contracting usually pushes those shut, unless there's damage to the valve itself or the chordinae tendineae. If there's damage to either one of those, then we could wind up with blood leaking back the wrong direction. Semilunar valves are closed during heart relaxation, but open during ventricular contraction. So these are closed when the heart's relaxed, but when the ventricles contract, they open, as opposed to the atrioventricular valves that close during that. Where atrioventricular valves are open when the heart's relaxed, they close during ventricular contraction. These valves simply open and close due to pressure changes. It's not like there's some electrical thing that causes them to open and close. It's actually the pressure changes within the heart that cause them to close. As the pressure changes, it forces those valves shut, and when the pressure goes away, the valves will open back up. Okay? The interesting thing about the blood inside of the chambers of the heart, those 4 chambers of the heart, is that blood inside of the heart does not actually nourish the heart. So the blood inside of the heart does the rest of the heart no good. So the heart's full of blood, but it gets no oxygen or nutrients from the blood that's inside of it. It actually has its own circulatory system. And this is where what we call the coronary arteries. And these coronary arteries actually branch from the aorta, and I'll show you how this looks here in a minute, to supply the muscles of the heart with oxygenated blood and get rid of the waste through the cardiac veins. The coronary sinus is a large vein on the back side of the heart that receives blood from the cardiac veins. So we have coronary arteries, and then we have a coronary veins and coronary sinus. So the coronary arteries are what gets blocked up in what we call a heart attack. When somebody has blockage in the coronary arteries, that's what's considered a heart attack. And that's where they go in there and they put those mesh stents inside of there to open those arteries up. A huge innovation. When I first got into EMS and first became a paramedic, I remember taking numerous people from a little small town in Oklahoma to Oklahoma City to Baptist Medical Center. They were the big heart place. That's where we went for long before we had the heart hospital or any of them places. We would take them to Oklahoma City, basically so they could see a cardiologist. The cardiologist would tell them, look, you're officially retired. Your life has forever changed because you have dead heart muscle, and your heart's not going to work as well as it once did. And depending on how much of that muscle was damaged would determine how greatly their life was impacted, or even if they survived. If it was a large amount of muscle, They didn't even survive because the heart couldn't function. Nowadays, it's amazing. We pick people up at their house. We determine that they're having a heart attack. We take them straight to the heart hospital or to a similar facility that's capable of taking a catheter, going in their femoral artery, all the way back up into the coronary arteries of the heart, finding the blockage and putting this mesh ladder framework inside of there to open that back up, and voila, we no longer have long-term dead heart tissue. A lot of these guys and gals are back at work within days, literally days. They're back at work. At most, they're off work for maybe a week, and that's just because maybe the job that they have is a really physical job, and they got to take it easy until the wound incision heals where they did the procedure. It's amazing, truly amazing, truly life-changing what it's done for people and how it's extended lives. But that's what's happening. That's what we do, what they call an angioplasty, where they go in there. Angiogram is when they go and look at it and find the blockage. Angioplasty is when they go in and they put that mesh stent. It's called a stent, but it's actually mesh, a mesh-looking thing. And it's usually an interventional cardiologist that does that procedure at one of our big heart hospitals or facilities that we have that are capable of doing that. Okay, so you can't see it on this particular picture, but remember, the blood inside of the heart doesn't actually nourish the heart. What it is this artery actually goes back up here behind this vein and ties in here with the vena cava. So this is where the blood comes from, behind that vein, and nourishes this artery down in here, and then it returns along these veins. And this artery actually wraps all the way around the back side of the heart and comes back up this way right here and goes back up this way, okay? And then this nourishes it. On the back side, we call the back side of this artery, I think they usually call it the circumflex, the right and left circumflex artery, where it goes around the back of that, if I am correct. But the interesting thing about this is the rest of the body gets its blood, even the lungs, when the ventricles beat, when the ventricles contract and squeeze, it pushes the blood. It pushes the blood out of these, and you can't really see that one, but it pushes the blood out when the ventricles contract. However, while the ventricles are contracted, the valve that's in here, the semilunar valve, actually closes off blood supply to the outside of the heart, okay? That's normal, it's supposed to be that way. So when the heart is contracted, there's no blood flowing through the coronary arteries. No blood flowing through these coronary arteries, at least no pulse, no blood pressure. But when the ventricles are relaxed, the back pressure, the pressure that's inside of here, and when somebody takes your blood pressure 120/80, that 80, that pressure, the bottom number of that pressure, so we got 120/80, is the amount of pressure that's in here when the heart's not beating. That pressure is a little bit higher right here, closer to the heart that we are, but that pressure actually pushes blood through the coronary arteries. And that's how the coronary arteries get their blood flow. Why is that important? Well, because if the heart's beating way too fast, if the heart's beating really, really fast, and it's spending so much time contracting and not so much time relaxed, then it's going to reduce the blood supply to the heart. Now the heart's working really hard and beating fast, but the blood supply to it's being limited. So what's the heart do to respond to a low blood supply or a low amount of oxygen? Usually it tries to beat faster. So we can get caught in a catch-22 if the heart gets beating too fast and we don't get it under control. So one of the reasons why we want to control a heartbeat when it starts to get too fast is it reduces the coronary blood flow, the blood flow that feeds the heart. The heart doesn't work, the heart dies, then everything else dies. If the heart doesn't get blood, eventually no organs are going to get blood. So there are a few exceptions to that because they do have those LVAD devices, the left ventricular assisted device that they implant in somebody that pumps blood through their body. It's kind of a weird deal, but it works. So those situations are different, but you would know about those when you come upon them. At least I would hope so. So, like I talked about here, this is just kind of giving you an idea of the blood flow through the heart. We've talked about that pretty in depth. So I don't really want to get all caught up in their wordiness of it. So one of the things that I want to talk about, and this really isn't in your book, but it's things that we've talked about. Remember, we talked about what happens to a cell. And I'm going to make you think back just a little bit, and I'm sorry, I'm not trying to pick on you, but remember when a cell is at its resting state, remember we've got a cell that has its sodium and potassium pumps have pumped out sodium, right? And pumped in potassium. But remember, it's pumping out 3 sodium for every 2 potassium, right? And because of that, we wind up with this cell membrane, or the inside of this cell. The inside of this cell is about negative 70 millivolts, roughly, or milliamps, or however they measure that. But it's roughly negative 70 compared to the outside. So there is a potential. Remember, we called that the resting potential, right? This thing's really kind of tough for me to write on. Of course, I'm not a good speller, so that doesn't help. So we got this cell. It's sitting there. We'll pretend like this is a cardiac cell. Okay? This is a cardiac cell. And it's sitting there at rest. It's waiting. So now we stimulate this with a little nerve impulse, okay? Remember, cardiac cells don't require nerve impulses. We're going to talk about that in a minute. But let's just say, for the sake of argument, this particular one, we stimulate it with a nerve impulse. We get this nerve impulse, a little action potential came down. It stimulated the membrane. Remember, we stimulate the membrane. What happens to the membrane? The sodium channels open up, right? Right? So these sodium channels open up, and what happens? Sodium rushes into this cell. So sodium's going to start rushing into this cell. So now the cell all of a sudden goes from negative 70 to maybe I don't know, positive, let's say, just for the sake of argument, it goes to a positive 10. So now we went from, say we're resting right here, and resting, this is negative 70, and we go to a positive 10, now we've got this, all of a sudden now we're up here to positive 10, right? And then remember, once those sodium channels open up at a certain time, the potassium channels are going to open up, right? And now some potassium is going to start to leak out. And now we start to go back to a negative number, and we may actually go as far as going to a negative 90. So now this is going to come all the way back down here, and now we're at a negative 90. So that's kind of what's happened in that cell, and then remember the sodium potassium pump. Sodium and potassium pump is going to kick in, right? It's going to kick in, and it's going to start pumping sodium back out and potassium back in, and it's going to get back to its negative 70 resting potential, okay? So now everything's going to go back up to here, and now we're back to negative 70, and we're ready for another electrical impulse, right? Hopefully what you're seeing me draw down here starts to look like something familiar, all right? Now, we're just talking about with one cell, what's going on. Well, if we group billions of these cells together, remember cardiac cells have that unique ability. They've got those intercalated discs, these little discs that touch the cells next to it, right? And each one of these cells are touching. So if we stimulate this membrane, it's actually, this disc is going to cause this membrane to be stimulated. All of it's going to happen simultaneously. All of these cells are nearly simultaneously, not quite, but all of these cells of the heart are going to contract, right? Now, remember though, we talked about the atria and the ventricles being separate. Now the ventricles are kind of tied together. The atria is kind of tied together, meaning if one of the atria fire, both of them fire. If one of the ventricles fire, both of them fire. But there is a true dividing line between the atria and the ventricles. The atria can fire and the ventricles may not. The ventricles can fire and the atria may not. It all depends on one little pathway. There's one little nerve pathway in there, and we'll talk about that in a minute, that connects the two. Otherwise, they're completely disconnected. So when I talk about cardiac cells firing, I'm talking about these cardiac cells that are grouped in a way that they can. So if this is cells of the ventricles, all of the ventricles are going to contract. And that's a large group of cells that contract. Or if it's the atria, it'll be all them. But that's a large group of cells. Remember, the ventricles are much, much bigger than the atria. So now we have all of those cells, and that's billions of cells contracting all at once. And that creates a lot of what's going on down here. It creates this on a big scale, a really big scale, something that we can measure. We can measure that electrical movement of these ions moving in and out of those cells when they've been stimulated, if there's enough of them, right? We have to have enough of them to be seen, okay? So if there's not enough of these firing off, we're never going to see it with what we typically read them on, okay? And I'm talking about a heart monitor. We put somebody on a heart monitor. This is what we're reading. We're reading the movement of these ions in and out of the cell on a grand scale, okay? We're going to talk about this a little bit more, so don't panic if you're still kind of trying to absorb it. We're going to talk a little more. All right? Remember I told you that we had a nerve impulse that set that thing in motion in the last slide? But remember that cardiac muscles have their ability to contract on their own. They have an internal set rate. But so cardiac muscles can actually be affected by internal factors or external factors, known as intrinsic or extrinsic. Extrinsic would be a nerve going to the heart. Intrinsic would be internally in the heart. Remember, the heart is actually innervated by the sympathetic and the parasympathetic nervous system. Either one of those two systems can have an effect on the heart. Remember that the parasympathetic nervous system is probably going to have an inhibitory effect, meaning it's going to try to slow things down. Sympathetic is going to have a stimulating effect to try to speed things up, and those two are usually in relative balance between the two, unless we have something happen that causes one or the other to take over. Eat a big meal, the parasympathetic is going to kick in, it's going to kind of slow things down. Go out for a jog, sympathetic's going to kick in and speed things up, especially if you're me, it's going to speed things up a lot because my body's going to be going, what are you doing, you idiot? So intrinsic and extrinsic factors are what affects the cardiac beating, a cardiac cycle is sometimes what we call it. The intrinsic conduction is called the nodal system, and this nodal system increases the heart rate and ensures that the heart beats as a unit. So we have these nodes, these areas inside of the heart, and they're areas of nerves inside of the heart, kind of like nerves, but it's a node inside of the heart that has the ability to tell the heart to beat. Either we can have an external factor that tells the heart to stimulate it to beat that's coming down from the brain, or if we don't get a signal from there, the heart has an ability to just set its own rate, which is a lot of what it does. It mainly sets its own rate. So there's different nodes in the heart that do that. The first node and the primary node is the sinoatrial node, or the SA node. It is the heart's pacemaker. It's the primary pacemaker. It is the main one in the heart. It's the node that should be the one that's making everything normal. If you've ever heard the term normal sinus rhythm, when somebody looks at a heart monitor, and it's because the rhythm they're looking at is coming from the SA node. The rhythm they're looking at is coming from the SA node, and that is normal, okay? It should come from that. That's what we consider normal. Something that I don't know if your book talks about, I can't remember, but the SA node has its own built-in normal rate. The normal rate for the SA node is 60 to 100 beats per minute, okay? That's normal. That's what's considered normal, and I know your book talks about this. So, but it is possible for the SA node to have a rhythm that's outside of that, slower than that or faster than that. If it's slower, If it's 59, we call that bradycardia, which means slow. Brady means slow. If it's over 100, we call that tachycardia. All right, so we call it tachycardia, which means fast. Just some terminology you should start to become familiar with. They will help you in any health career that you're planning on taking. So the normal rate of the SA node is between 60 and 100. That's normal. Below that is bradycardia. Above that is tachycardia. Okay. Well, I'm not going to get off into the weeds about how to tell which rhythm is what you're looking at. This isn't a rhythm interpretation class, because if it was a rhythm interpretation class, we would spend about 48 hours together, literally, or more, going over all of that stuff. This is just actually what you need to know before you take that rhythm interpretation class. The atrioventricular node is a node that's located at the junction of the atria and the ventricles. Remember, I told you there's a pathway that connects the two. There's a node right there in that pathway, and the atrioventricular node is a junction, or the AV junction, sometimes that we call it. Oftentimes we call it a junction instead of a node. It also has its own normal intrinsic rate, which its normal rate is 40 to 60. Now, I'm not going to talk to you about how we know that a rhythm's coming from the AV node versus a sinus node versus a ventricles or anything like that. I'm just telling you this is normal for sinus, this is normal for ventricular, and no, there's no such rhythm as a normal ventricular rhythm. We just don't call it that. A ventricular rhythm is not normal. So, and then we get down to the AV node, or the atrioventricular bundle. It's also called the bundle of His. It doesn't have an intrinsic rate, but it is the branching. It's where that kind of branches out, and then it's the area where these branches right here will split, and I'll show you that. I believe the next slide shows us that. And then we have these Purkinje fibers, and they're right there at the wall of the ventricles, and they're just like the finger-like attachments of those nerves right there. And these down here have a normal rate of 20 to 40. So that's just their normal intrinsic rates. These are not things that I'm going to test you on, other than you probably should know what bradycardia and tachycardia is. That would be important, but I'm not going to test you on what the intrinsic rate of the AV node or the Purkinje fibers are. I just wanted to provide you with this information for some context, to maybe help you kind of understand. When we look at a heart rhythm, we're looking at this heart rhythm, and the normal rate for certain things comes into play. It becomes a factor when we're trying to determine what that rhythm is and whether it's normal, abnormal, what's causing it, and those kinds of things. This is part of that information that we use. It's not all of it, but it's just part of it. So here, this is a really good picture of the nervous system of the heart. So we've got this node, this SA node up here, and notice that there's nothing connecting these two nodes. What happens is this node is going to fire off, and these cells, these cardiac cells that are close to this, that impulse is going to spread out. Remember the intercalated discs between these cardiac cells? It's going to cause this to spread out over the atria and over the atria on both sides, right? But the only connection between the atria and ventricles is this right here. This is the only connection. If this doesn't pick up the impulse right here, then it never travels this way, okay? This right here is the AV node, okay? This little blob right here is the AV node. Right down here is the bundle, okay? These are the bundle branches, one on the right and one on the left. If you've ever heard somebody speak of a bundle branch block, that means one side of this is actually conducting the impulse slower than the other. And then down here at the bottom, these are the Purkinje fibers. These little fibers down here, these are the Purkinjes, okay? And you'll notice how these nerves, you see how these nerves go down and then they come back up? They go down this way and come back up, down this way and come back up. Why is that? Why do you think that is? Well, I'll tell you why that is. because it works better if the ventricles start squeezing from the bottom first to push the blood out. So we want the ventricles to squeeze from the bottom and then back upwards. We want them to actually squeeze backwards. Where the atria, we want them to squeeze normal to push the blood down. We want these guys to squeeze backwards to push the blood out, okay? We want them to squeeze from the bottom up so it pushes that blood out this way, right? And you can't really see where this one's going out this way, but we want to push from the bottom up. And that's how the conduction system, the electrical conduction system of the heart, remember that's the intrinsic one of the heart, that's how that works. And that's going to become more and more important when we put somebody on a heart monitor and we start to look at things and try to understand what's going on. All right, so normal intrinsic rate, conduction rate's roughly about 75 beats a minute, so that's normal, but it's different for all of us. Contractions are usually initiated by the SA node. Not always. We can have some problems within the heart, but in a normal, healthy heart, the SA node is the one setting the pace. And then, of course, we have the other cells sequentially stimulated. And then, of course, the cardiac muscles depolarize in one direction from the atrial to the ventricles, and the ventricles push it up and out, just like we looked at. Once the SA node starts, that impulse is going to spread, and then it's going to make the atria contract. At the AV node, the impulse passes through the bundle branches and Purkinje fibers. The interesting thing about the AV node and the bundle, it'll actually pause that electrical impulse for just such a millisecond or a microsecond or however small of a second it is. It'll actually pause that and give the atria a chance to fully contract before it sends that impulse on, because it's important that the ventricles are completely full. So another reason for the AV node and the bundle is to slow that impulse between the atria and ventricles. It pauses it for just a little bit. It'll give it a little bit of a pause. And then, of course, blood is squirted out of the ventricles and in the pulmonary tract and all that stuff. So when we put somebody on a heart monitor, it's important that we put the leads on in the right place. Remember the direction. Remember what I showed you, that the heart sets tilted? Remember, and I'm a horrible artist, so we'll just say, and this really does look like a heart. Well, I'm not really trying to make it look that way, but remember the heart's kind of tilted that way, and it's kind of on this axial plane going this direction. So when we put the heart leads on somebody, we put the negative lead up here, we snap it on them up here, we put the positive lead right here, and most of the time we put our ground right here. Some people will put the ground down here, not absolutely critical, but this setup right here is what we call lead 2, okay? So that's the lead 2 setup. No, this is not anything that I'm going to test you on, just something that I wanted to point out to you so you kind of have a little bit of an aha moment. Now, remember, SA node up here, right? Notice that this is negative and this is positive. If we drew a line like this and we said anything that moves towards this, so if we have anything that's moving towards this, any electrical energy that's moving towards that, moving this direction, will go this way, okay? All right? Anything moving away from it, going the opposite way, will show this way, okay? All right, so hang in there with me. We have the intrinsic SA node fire off, right? And it sends that signal out through the atria, right? Both atria sends that signal out. But remember, the atria are not real big, not a lot of muscle there, so it's pretty small. And this is actually going this way, right? You can see that that impulse is actually traveling towards the positive, but since it's not really a lot of let me look first my eraser. Since it's not a lot of muscle that's involved, we're only going to see a little small blip, okay? That's all we're going to see. That's all we'll see from that. We'll actually have this AV node right here And then this kind of comes down this way, branches and goes like that. And all right. So just excuse my crudeness, but we have the AV node right here, and when that impulse gets to the AV node, remember the AV node is going to kind of hold it for a little bit, so it's going to give it a little bit of a pause right here, and then it's going to send it on. And as it sends it on down this way, down both of these bundle branches, it won't be able to tell the difference between right and left. It just won't. Both of these impulses are traveling at the same speed in this direction towards the positive, but it's a lot of muscle, right? A whole lot of muscle that's being contracted. All of a sudden we see this spike go up like this. It gets down here, makes the turn, goes this way, and now, now it's going away from the positive lead. So now it's going to go back down here, but it's a big impulse, right? And then as this all settles back down, this is going to want to come back up to normal, back up here to normal, right? So now this is where we're at, okay? So what's happened inside of here is all of those cells depolarized in that area, all of those ions changed place, and it gave us an electrical charge of this group up here that was moving in this direction that gave us this blip right here, which this blip, sorry, that's pitiful, horrible. Come on, let's get that off of there. Which this little blip right down here is what we affectionately call the P wave. And it has nothing to do with going to the bathroom. It's just called the P wave. And the P wave is atrial contraction. Okay? So that's what's going on with the atrial contraction. This one down here, this holds it right here, and we call this this little pause right here, we usually call this the PR interval because it's the space between the P wave and this wave right here that is the R wave. Sometimes there's a little wave that goes backwards down here, and we call this the Q wave, and then this wave down here is the S, and then this is we'll get to that in a minute. So anyway, so now this impulse travels on down here. We've paused it right here. We've got the PR interval, okay, with this little pause. Now this travels on down this way. We see the little Q. Sometimes we'll have this little dip there. We don't always see the Q wave. Most of the time we don't, but usually the Q wave's there. It's just that we can't see it. Q, R wave going up, S wave right here going down, and then this part coming up is oftentimes we call it the ST segment. We'll see a little blip right there. And the reason why we see that blip is, remember when the sodium and potassium pump kicks in? Excuse me, and it starts pumping sodium and potassium back, into and out of the cells. Remember that movement of ions that resets that cell to its resting potential of -70? That's what this is right here. It happens up here in the atria, that reset, but we usually can't see it because it's buried somewhere in here. And some of this here is actually buried a little bit in here. What we're seeing a lot of here is kind of some of this part of the ventricle. So that is where a heart rhythm comes from. And when we look at a heart monitor, that's what we're looking at. And of course, what I showed you was what we look for in a normal PQRST. This is the ST segment, or just the T segment. So that's what we look for when we look at a heart monitor. And if we know what normal is, if we know what normal is supposed to look like, then we can start being a little bit of a detective and finding out what's not normal, what needs to be fixed, what needs to be corrected, what's not working right. So that's a PQRST. That's it in a nutshell. And that's all I'm going to talk about when it comes to QRSs and ECG and heart rhythm. And actually this right here, what we look at right here, this atrial ventricle contraction and reset, is a cardiac cycle. Oh my gosh. So that is one cardiac cycle, okay? I told you don't make fun of my spelling. So that's what that is. That's what's going on there. And I wanted you guys to have an opportunity to look at that in hopes that it would help you understand that. Remember, this is why I talked so much about the movement of ions across cells and all that kind of stuff, was so that when we got to this, you would go, hey, that's pretty doggone cool. And hopefully it'll make it to where that you understand it better when you see it later, when you further your education. This slide here, actually, I was planning on putting a lot of what I put here on this slide here, so that's why there's another blank spot in there, so don't sweat that blank spot. Heart contractions, there is if we do have an imbalance in our contractions, we can have various different things that can happen. Why this thing picks heart block as something to talk about, I don't know, because it's not the only thing that can happen to a heart rhythm that can affect what the heart does, the rate of the heart, how fast it beats, slow it beats, what it does, all that. But this talks about a heart block. Heart block is basically damage to that AV node, the node between the atria and the ventricles. Depending on the severity of that, we may actually have the SA node kind of firing and doing its thing, but it's not getting past the AV node, so the atria is firing, but the ventricles aren't responding. So there's kind of a block, and that block can be of varying degrees. It can be completely blocked, partially blocked, quite a bit blocked. So there's different levels of blocks that we can talk about. We have a first degree block, a second degree. There's two different types of second degree blocks. There's a third degree block, which is the complete heart block. So what these do is it actually affects the ventricular rate, how fast the ventricles contract, and it also affects how much blood actually fills the ventricles. Remember, the atria are responsible for topping off the ventricles, and if those two aren't in sync, the ventricles are going to be pumping without having been filled up completely. There's several other different types of rhythms out there. That's just an example of one. There's also fibrillation, which is rapid, uncoordinated shuttering of the heart. We can have fibrillation in the ventricles, which is bad and usually means death if untreated, if we don't shock them. But we can have fibrillation in the atria that a person can live with for years, 10, 20, 30 years, as long as they're under a doctor's care and they're treated properly. The problem with atrial fibrillation, whereas ventricular fibrillation means death if not treated immediately, ventricular, or if we don't make the ventricular fibrillation stop, if we don't correct it. Atrial fibrillation usually can't be corrected, but if it's not treated, what happens is, you remember we talked about platelets in the last chapter, or when you read about platelets in the last chapter, if we have a motion that's causing those platelets to kind of smash together and agitate them, they'll start to clot and start the clotting factor. If we have atrial fibrillation, it causes like a butter churn inside of the atria, which can start the formation of blood clots, and those blood clots will then travel to different parts of the body, brain, the heart, those kinds of things, where they can be very, very problematic, causing strokes. So that's why somebody with atrial fibrillation needs to be treated on a regular basis by a doctor, and they're probably going to be on some blood thinners and those kinds of things, and maybe even something that controls how fast their heart beats, because with atrial fibrillation, the heart can actually beat too fast. So ischemia is basically a lack of oxygen to the heart muscle, usually caused by either a partial or a complete blockage of the coronary arteries. Remember those arteries that we talked about that feed the heart? Ischemia, infarction. Infarction means that we have a complete block, and it's not inadequate, it's a complete loss of blood supply to an area of the heart. So untreated ischemia oftentimes leads to infarctions, but all those can be treated typically with stents, or where they go in and do what's called a CABG, a coronary artery bypass graft is a CABG, where they do basically, you hear them talk about double, triple, quadruple bypasses, where they go in there, they bypass those arteries with arteries they've taken from the patient's leg. So Tachycardia rate, any heart rate over 100 is tachycardia. Any. It doesn't matter where it's from, even if it's, I said that a ventricular rate of 20 to 40 is the normal rate for a ventricle to beat on its own. It's not normal, but it's what it does on its own, the Purkinje fibers. But in order for that to be considered ventricular tachycardia, it has to be above 100. So the origin of the impulse is from the ventricles, and it's over 100. That means it's ventricular tachycardia. If the origin of the beat is from the junction, then and it's above 100, it's junctional tachycardia. So now the only thing we usually call bradycardia is sinus rhythms from the SA node. If it's below 60, we'll call it bradycardia. From the other places, don't usually call it whatever bradycardia. I don't know why, they just don't. Just save you a little heartache when you take your first EKG class. You don't go, hey, that looks like ventricular bradycardia. Well, no, there's no such thing. Or maybe there is, or there should be, but there isn't. So just so you know, tachycardia rate over 100, and that's very true with the SA node. Bradycardia rate below 60, and only with the SA node. All right? Cardiac cycle refers to one complete heartbeat, a systolic contraction and a diastolic relaxation. About 75 beats per minute is normal, and the length of a cardiac cycle is about 0.8 seconds, so just under a second. So if you see a test question on that, it's just under a second, 0.8 seconds. 0.8 second or seconds, however you say that. All right? These next few slides start to talk about different heart sounds in the cardiac cycle and how they're related. I did not put any questions in there about heart tones and heart sounds, just because those are very, very difficult to distinguish, even for regular physicians. Cardiologists spend their career getting good at those. So for us to think that we can do that in a classroom is a little silly, or even online, especially online. During mid to late diastole, the pressure is low in the heart. Blood flows into the atria and the ventricles. The semilunar valves close and the atrioventral valves open. The atria contracts and it forces the blood in the ventricles. And during the ventricular systole, the blood pressure rises as the ventricles prepare to contract. The atrioventricular valves close, causing the first sound that we hear. Those valves closing give us the lub sound. And then the semilunar valves open as the blood pushes against them. Blood travels out of the ventricles through the pulmonary trunk and the aorta. The aorta is relaxed and filled back with blood. The second heart sound is heard when the semilunar valves close, causing the dub. That's at the end of the systole. All four valves are briefly closed at the same time, and that gives us the dub. So the reason why I went ahead and left this in here to talk about is it's a very good indicator for how well the heart valves are working. They will listen to your heart valves in your heart and try to see how well your heart valves are working. My doctor actually does this as a routine thing every time that I see him now, because my father actually had mitral valve issues. He still does. And they heard his mitral valve malfunctioning listening to it with the stethoscope. His cardiologist did. So now they listen to mine and my brother's heart valves just in case there's something going on with them that can be fixed or prevented from getting worse. So that's why the lub dub sounds are important, and that's what they're listening to. It's the quality of the lub dub sounds that they're listening to, or if there's a little swoosh or a little something going on in there. Like I say, I do not expect you, nor should you even begin to expect yourself to be able to listen to heart sounds and go, that's normal, that's not. Just because some physicians are not very good at it. As a regular doctor, that's not something that they do teach them that, but it's not something that they routinely do. Cardiologists are very good at it. Some internal medicine docs are pretty good at it because they've done it a lot. It's something that you would have to do thousands of times in order to get good at it. So don't think that you're going to be able to go out and diagnose heart valve problems by grabbing your handy dandy stethoscope and going out and listening to people to save the world. So it's a nice thought, but probably not going to work out. And of course, faulty valves produce sounds or whooshing sounds that are called murmurs. They're abnormal sounds, and that's actually what those guys are listening for when they're listening to that lub-dub. They're listening to try to catch a murmur to find out how well or how poorly a valve is working, and usually that results in more tests. They don't typically diagnose a bad valve just by listening to it. If they're concerned about it, they order more tests. Okay, so cardiac output is the amount of blood pumped by each side of the heart in 1 minute. The way we measure that is first we have to determine the stroke volume, which is the volume of blood pumped by each ventricle during each heartbeat. Normal is about 70 mils out of the left ventricle. And then we take the heart rate, which if a normal heart rate is 75 minutes. So we take the stroke volume, multiply it by heart rate, and that will tell us what the cardiac output is because that's all measured over the course of a minute. Stroke volume, cardiac output, stroke volume times heart rate will give us that, and we can see the math here when they do that. About 5.25 liters of blood per minute, or roughly your total volume, or actually a few times your total volume in a given minute circulated around. About 60% of the blood in the ventricles is pumped out with each heartbeat. Now that's going to change depending on needs and demands and all that kind of stuff, but and everything being normal and not under stress or anything like that, about 60% of blood is pumped out, about 70 mils. Still some left in there. And then there's this Starling's law of the heart. It plays a critical role in Starling's law, but it actually points out the critical role of controlling stroke volume. The more the cardiac muscle is stretched, the stronger the contraction is going to be, at least to a certain degree. So if we stretch that and force more blood into the ventricle, it's going to cause that muscle, the cardiac muscle, to give a stronger contraction. The problem with that is that stronger contraction, if we're called upon to do that for long periods of time, just like any other muscle in the body, the heart muscle is going to start to get bigger if it's stressed like that. So someone who has uncontrolled blood pressure or other cardiac related problems and they undiagnosed, this issue is allowed to go unchecked, might wind up with an enlarged heart, cardio hypertrophy. We see young kids with this, and it can either be fatal if they're a very athletic child, and it's probably in part because of their athleticism, or it can be career-ending. Basically, you're no longer an athlete because you need to settle down. What happens is when there's much more heart muscle to be had, there's many more vessels going to the heart and much more risk of one of those vessels being blocked. You have more vessels, you increase the risk of a block. So that's the concern with Starling's law. So I talked about this a little bit earlier. We can modify the heart rate with a neural impulse from the sympathetic or parasympathetic nervous system. Also, some hormones and ions and even some physical factors can do it. Remember, there's some hormones, we can release some chemicals like epinephrine into the bloodstream, and it will cause the heart rate to speed up. So neurally, we can have hormones and physical factors, and then the heart has its own intrinsic factors that kind of make it beat. But usually it's extrinsic factors that cause it to speed up, sometimes cause it to slow down. Okay? Just left this slide in there so you can kind of see how all of that ties in with, I'm not going to go through all of it, but and how that affects cardiac output, how much blood is output in a given minute. So, blood vessels. All right, so enough about the heart. The heart would be absolutely of no use to us if we didn't have something that could carry the blood away from the heart into the tissues. So we've got this vessel system, this vascular system that does that. Arteries and arterioles carry blood away from the heart. Vessels and venules carry, veins and venules carry blood back to the heart. In between them, usually we have capillaries where we have where blood and oxygen and other nutrients are exchanged at the tissues in those capillary beds that are in between veins and arteries. When we look at a vessel, we can take a cross section of a blood vessel and break that down into 3 different layers, or tunics as they're called. The tunica intima forms a friction-reducing lining of the endothelium inside of it, inside of the blood vessel, so that inside lining where the blood passes through. The tunica media usually has some muscle properties to it and some elastic tissues. This muscle that's there is controlled by the sympathetic nervous system, by the sympathetic nervous system. And then the tunica externa is the protective outermost covering that kind of has some connective tissue that holds the veins and arteries in place. But that's the thing that they have in common. We can see the difference between the two. That muscular layer of the two is vastly different between an artery and a vein. And we can see that here. This is all the muscle of the artery, and there's not much muscle here in the vein. The reason for that is these arteries are under a lot of pressure from the heart. The heart has them under a lot of pressure, forcing blood through them, so they're expanding. They're getting bigger and smaller and bigger and smaller. So it's like a workout for them. So the muscle gets a little bit bigger inside of there. Also, that muscle can be used to increase blood pressure or decrease blood pressure by constricting or dilating, make the vessels bigger or smaller, which is controlled by the sympathetic nervous system that controls that. So looking at the two side by side, you can see that arteries are much more muscular, and veins have much less muscle in them. But veins, the interesting thing that a vein has that an artery does not is, well, maybe I won't find my pen, is veins have valves. So these valves actually prevent the blood from flowing backwards. It keeps blood moving in the right direction. Remember what I said about valves? Arteries don't have valves in them because the arteries have a constant amount of pressure in them all the time. Veins may have a constant amount of pressure, but that pressure is low, and it wouldn't prevent blood from backflowing. So it's important that we keep the blood from going backwards. So the arteries have that thicker middle layer of muscle that helps withstand changes in pressure, whereas the veins, that middle layer is much smaller. Also have those valves that prevent backflow. One of the things too is you see the skeletal muscles on either side of this valve. The skeletal muscles actually help, these skeletal muscles here actually help by, when we're up walking or moving or that kind of thing, the pressure that it puts actually helps push the blood back towards the heart. These valves keep it from moving backwards. So if we contract our muscles, it'll push the blood in only one direction. The valves keep it moving that direction. It's kind of a way to assist moving blood back to the heart. It's especially true in the muscles of the legs and those types of things. And that's why if you have poor circulation, it usually affects the legs, like diabetics. It starts to affect the legs, and eventually the circulation may get so poor that they wind up having a limb that dies. The tissue in the limb dies, and they have to remove the limb. All right, so at the capillary level, these things are only one cell layer thick. The reason why is so that it can exchange those gases between the tissues. These capillaries also form networks that we call capillary beds. And the blood flow through these capillary beds is referred to as microcirculation. So remember, while arteries and veins are relatively thick and arteries are very thick, at the capillary level, these things get down and they're very, very thin to allow nutrients to be exchanged. Capillaries do have the ability to shunt blood through them. They do, where they can actually control the direction, or not the direction, but how much blood actually goes into the capillary bed as a whole, or it may just pass straight through it, depending on what's going on in the body. and it controls that through these precapillary sphincters. And it can control the flow to certain tissues or something like that in the body based upon the needs of the overall body. We can see these here on this diagram. You see these right here. These can tighten up and cinch off and keep blood from flowing in these directions and only allow it to flow through there, and instead of the whole circuit like this. That's what it shows here. Keeps the blood from flowing in that direction. Varicose veins are basically a structural defect because of bad valves. It's a common problem, especially people who are overweight and people that are on their feet all the time. There's some predisposing factors from that, but don't think just because you have varicose veins you got some weird, crazy abnormality going on with you and that you need to rush off to your doctor. Most of the time, it's not all that unusual. So, largest artery in the body is the aorta. It's the one that leaves from the left ventricle of the heart. Of the aorta, you have the ascending aorta. It's the part that is going up. It's going towards the head, so it's the right when it leaves the left ventricle, it's ascending. Then you have the arch, the hump of it, and then the thoracic aorta, and then the abdominal aorta as it passes down through the body. This is what they're talking about, this aortic arch here at the top. This is ascending, and this is the arch, okay? And then it's going to be descending, but then it's going to get into the thoracic area where they call it the thoracic aorta. You see the arch right there at the top of the heart. You see the ascending, the arch, and then it goes into thoracic and descending, and down in here, and it's the abdominal aorta, and then it branches down here. It is possible for there to be an aneurysm on this, or to develop an aneurysm on that, where the musculature of that begins to fail, kind of like a, oh shoot, I started down that road and I can't remember what I was going to say. Anyway, it happens in the stomach. I can't think of what it's called. But you can have that musculature fail and it starts to develop a bubble on it, which is called an aneurysm. And it's called an abdominal aortic aneurysm. They're not horribly common, but they do happen from time to time. And there's some key indicators that you can look for. I've treated a couple of patients with those, and they can be pretty frightening. All right, so then we have the right and left coronary arteries that serve the heart. We talked about those. Then the aortic arch, that branches off into a couple of brachiocephalic splits to the right common carotid artery and the subclavian arteries, and then the left common carotids, the left subclavians. It branches out, intercostals and all that kind of stuff. And I realize this is branching out, and I'm really just skimming through this because it's not something that I want you to worry about the exact direction and how these break down and how they split and all that kind of stuff. I'm not going to test you on that. It's more important that you understand how these arteries and veins work and what they do and what they're made up of than it is for you to be able to label which ones are where and what they do. So there's the heart. We've looked at that, and we can see the veins here, and then the whole network of veins. There is a couple of them, though. I'm just backing up. Sorry. I would like to point out that are noteworthy. The femoral artery right down here. This femoral artery, the radial artery, and of course there's the brachial artery that's up here. The brachial artery is the one that when you're taking a blood pressure, you're putting your blood pressure, your stethoscope right there. The brachial artery is the one you're listening for the blood pressure in, so that's why I'm pointing that one out. The radials down here is one where they routinely do blood gases, so they'll get blood from that one right there. The interesting thing about that one, it runs right along a nerve, so blood gases can be painful. The femoral artery is another great location, kind of like the carotid artery, for us checking for a pulse. Because we can feel a pulse down here when somebody's blood pressure's really low, like 60 or 80, or below 80, we can feel it at 40 to 60. Also, this is the location, one of the locations, actually the main location, where they put that catheter in and they run it up here, back up into the heart to look at the vessels of the heart and to put stents in it. So usually in one of these two locations, they're actually starting to do it a little bit with the arteries too, or the radial arteries as well. So I wanted to point out those. If you know those, ironically enough, the vein that runs right next to that artery has the same vein or the same name. So it's easy to know the name of a vein if you know the name of the artery. So that's pretty easy. Usually the nerves is the same way. So all of these veins return back. We have, in the vena cava, we have a superior vena cava and an inferior vena cava. And where I'm talking about is, inferior vena cava, superior vena cava. This brings blood from the top of the body, blood from the bottom of the body back to the heart. It's just a returning of the blood back to the heart. And this is going through all those veins that it goes through to do that. If I would have realized it, I would have took these slides out. My apologies. It brings them all back. This does talk about, where's it at? There's one that I want to talk about. Oh, this right here. Wait, where's it at? Where's it at? Sorry, guys. I'm looking for something. Oh, that's it. I was looking for it. This right here, the circle of Willis. I always like to point this out. You know, I told you about the loop of Henle in the pancreas where the A and B, where insulin and all that's produced. Well, this is the circle of Willis, and it's in the brain, and it's this loop right here in the brain, and it's just a blood flow loop that consists of all of these things right here. I don't know why I like to point that out, and maybe just because it's kind of neat. It's named after somebody that I guess discovered it. Named after somebody probably, I would assume, named Willis. If you're wondering what you're talking about Willis, they're talking about the circle of Willis. Sorry, couldn't resist. Okay, there is a little bit of circulation that I want to talk to you about just a little bit, and it's called this hepatic portal circulation. The interesting thing about the hepatic portal circulation, you know, arteries go down to the tissues and veins come back. The interesting thing about the veins that come out of the digestive organs and the spleen and the pancreas, what's interesting about the veins that come from them, they all go through the liver first before they go back to the heart. The liver is part of that filtration system. So any of those nutrients that are absorbed in the digestive system, and then different things that are released by the spleen and the pancreas, are picked up by the liver before they go into the rest of the body. So it's kind of a filtering system to keep bad things out of the body. So, and the reason why I point that out is if somebody drinks alcohol, they drink alcohol. Alcohol is actually, the body sees alcohol as very toxic. So it is immediately absorbed by the veins around the stomach and goes straight to the liver, right? So this is why alcoholics eventually over long periods of time begin to damage their liver, because that alcohol is actually toxic to the body, is absorbed in the stomach and quickly goes to the liver. The liver can't keep up with filtering it out, and it starts to damage the liver and those types of things. But anything, you can see anything that we absorb down here in any of these actually is filtered through the liver first before it goes back to the heart. So nice big filter. And then this talks a little bit about fetal circulation. Fetal circulation is a little bit different. I'm not going to get real crazy about testing you on fetal circulation, but I do want you to be aware that there are some differences in fetal circulation. So we've got the placenta down here. And the baby's receiving nutrients from the placenta, right? And it's returning waste products back to the placenta. So this artery that's going down this is actually plugging into the vena cava. It plugs into the vena cava, so nice, nourished, rich, oxygenated blood is coming in through the vena cava and all that. Remember, the lungs aren't working yet, still inside of mama, so the lungs aren't getting oxygen. So the oxygen-rich blood is actually coming from the inferior vena cava. It's returned back through a different system um, through the veins, the arteries actually of the lower part of the body, it kind of loops back up. So different circulation. What causes these to close off is when the baby's born and they kind of take their first breath. That pressure causes this, things to close off in the heart. The pressure causes this to close off. That fetal circulation goes away, and they start to rely completely on their own body's circulation. Amazing how all that bypassing happens. And this goes into that. I'm not an expert on that, I'll be honest with you, of exactly how that happens, but basically the flow of blood completely bypasses the lungs and the lungs and the heart in the baby while they're in there. So it's interesting how all that works while we are still in our mother's womb, and then when we're born it changes. So it's pretty neat how that works. So places that we can check for a pulse. These are very common places to check for a pulse. We do check a lot of times. You may be thinking, why do we need to check a pulse down here? Why would we need to do that? Well, sometimes if we have a limb injury or something like that, we need to check for circulation. Checking for a pulse there is pretty good because even if the blood pressure is pretty low, we can get a pulse there. We can feel a pulse up here, even if their blood pressure is as low as 60. We can't feel a pulse if their blood pressure here, but if their blood pressure is below 80. So around 60 or so, we can still feel it in those two areas. So that's why sometimes we check. If you have a healthcare provider that you see check a pulse there, you're like, you may be thinking, what is that weirdo doing? Well, what they're doing is they're checking for a pulse. It's actually pretty normal for those of us in healthcare to do that. We don't always think what other people think when they're watching us do it. So I'm betting most of you guys are pretty familiar with the concept of a blood pressure, but basically it's the pressure that's exerted against the walls of the vessels that's caused by either the pressure of the vessels, the size of the vessels, or the beating of the heart. So basically what happens is you have two numbers. You have the 120 over 80 numbers, right? 80 is the constant pressure inside of the arteries that we're measuring. The 120 is the pressure being exerted by the heart every time it beats. So every time the heart beats, it's actually raising that pressure 40 points. That's in a nutshell what's going on. Why is that important? Because when we look at somebody's blood pressure and we say their blood pressure is 210 over 110, well, now we know that the amount of pressure in their arteries without the heart beating is 110. That's a lot of pressure. And now we see that the heart is getting up to 220 or whatever. Now we know the heart's working really, really hard too. Sustained like that, we're going to have all kinds of problems where it's going to cause the artery walls to get thicker because of the pressure on those artery walls. Remember, if those artery walls get thicker and thicker, they're going to grow inward, and the space in the middle of the arteries is going to get smaller. It's going to increase the likelihood of heart attacks and strokes. Also, it's going to thicken the walls of the heart and increase the likelihood of a heart attack. So there's all kinds of problems that can happen there. Of course, we measure a blood pressure with a blood pressure cuff and a stethoscope. There is more than one way to do that, but the only way we'll get an accurate blood pressure is with that blood pressure cuff and a stethoscope, unless we're using an automatic blood pressure cuff, which is pretty common nowadays. When I first got in this business, there wasn't such a thing. Or if there was such a thing, nobody can afford them. Now they're everywhere. So I'm assuming all of you guys know how to measure a blood pressure, but we measure it with the brachial artery. It's best to measure it on the left side of the body because that's the side that the heart's on. Of course, if they've had a mastectomy or something like that, or if they have a shunt in there for their diabetes, we don't want to use that side. Or if they've had a shunt there in the past, we don't want to use that side. We can actually take a blood pressure in somebody's leg, in the big fat part of their leg, in the thigh part. So sometimes we have to do it that way too. We do need to notate if we take the blood pressure in a different place than what we normally take it. So we pump the cuff up until we've occluded the blood flow. We can't hear anything. And then we gradually let the pressure out. Listen for the first noise that we hear, and that is the top number. And as we let the pressure out, when that sound changes, that's the bottom number. Easy as that. So, so the amount of blood pumped out plus peripheral vascular resistance equals blood pressure. Just a really wordy way of talking about blood pressure. So, the sympathetic nervous system can cause the blood vessels to narrow, cause something called vasoconstriction. Vasoconstriction actually will increase the blood pressure. The body's going to do this for a various number of reasons. Most of the time it is well needed when it does do it. Sometimes it may be something else going on. The kidneys can also regulate blood volume by either holding onto water or letting go of water, and the way that it does that is the use of renin, which is an enzyme. When the pressure in the kidneys is a little bit low, it'll release renin, which will trigger the formation of angiotensin II, which is a good vasoconstrictor. What's interesting is angiotensin II stimulates the release of aldosterone, and that stimulates the sodium reabsorption, which causes the body to hold on to water. Also, angiotensin II is a vasoconstrictor. It'll cause the vessels to constrict. There is an enzyme that actually converts angiotensin I to angiotensin II, and renin plays a role in there. But we can take a medication that is a medication that blocks that will block the enzyme from converting angiotensin I into angiotensin II. Angiotensin II actually causes not only sodium reabsorption, but it also causes vasoconstriction. That medication that we give is called an angiotensin converting enzyme inhibitor, or an ACE inhibitor. So that is really the focus of a lot of the medications that they give initially for blood pressure are ACE inhibitors that inhibit the conversion to angiotensin II, which will control the blood pressure, keep the blood pressure from going up. Like I say, sometimes the body resets some of the things in there and it thinks things should be higher. A lot of genetics going on when that happens. So temperature also will cause vasodilation. Too hot will cause vasodilation. Too cold will cause some vasoconstriction, so that can affect the vascularity. Chemicals, substances that can increase or decrease blood pressure. Epinephrine increases heart rate. There's all kinds of good drugs and bad drugs out there that can affect the diameter of the blood vessels, the rate at which the heart pumps, and all that kind of stuff. Quite a few things out there. And of course, diets low in salt, saturated fats, and cholesterol helps prevent hypertension, but there are some predisposing factors that you could be a very, very healthy eater and doing things right and still have high blood pressure. It's entirely possible. Or you could eat like a pig, and still be lucky to be skinny and dump salt on everything and, live to be 105. Obviously there's healthy ways to live. I would venture to say that most people that live very, very badly, that don't take care of themselves like that, are not so fortunate. It doesn't end well for them. But you always hear about those few, and then you have people that think, well, if so-and-so can live that way, I can. Well, it doesn't work that way. All right, so normal range for blood pressure, 140 to 110 for systolic, and then 70 or 80 for diastolic. That's pretty much the normal range. When we get outside of that range is when we start to classify some problems. Hypotension, which means low blood pressure, is a systolic. That top number is below 100. Usually there's some illness going on, but sometimes it could be other factors. High blood pressure, on the other hand, if we have a blood pressure above 140 with the top number or above 90 with the bottom number, that's considered high blood pressure. Either one of them two numbers is above 140 or 90, then we call it high blood pressure. Actually, blood pressures between 80 and 90 is considered pre-hypertension nowadays. They don't treat pre-hypertension, but they start to watch it a little bit more closely. All right, so gas exchange and nutrient exchange. Remember, these exchanges happen based upon concentration gradients. From higher concentrations to lower concentrations, there are some pressures that affect this, some hydrostatic pressures, some pressures inside of vessels that affect this, but mainly it is purely done through concentration gradients. If you've got more oxygen on one side than there is on the other, oxygen's going to diffuse from one side to the other. So as oxygen-rich blood reaches the capillaries, that oxygen is going to diffuse out of the bloodstream, and the bad products are going to diffuse into the bloodstream because those concentrations are different. Since there's more carbon dioxide in the tissues because of cellular metabolism, that carbon dioxide concentration is much higher than what's in the bloodstream of the blood passing by. It's going to pick up that carbon dioxide and those other waste products. Remember, it's all due to these concentration gradients. There's no magical thing in there, telling people which direction to go. You don't have a gas exchange traffic cop in there saying, well, you need to get off and you need to get on. It's all done by the concentrations that are in there that create pressure changes or differences in pressures. And the reason why I keep throwing pressure differences in there is the oxygen molecules actually have the ability to exert pressure, exert force because of the way they move. And the more of them that there are, the greater the pressure that it's responsible for. So, and that's where that concentration gradient comes in. So these substances are going to move across, are going to change places in various different ways, either directly through the membranes. They may go through some intracellular clefts, through pores in some of them capillaries, or through even vesicular transport. You know how some of these cells have these vesicles that they transport stuff with. So various different ways that things are transported through these capillaries. So it's not just one route, right? In capillaries, it depends on the substance, will determine the route that it's going to take. All right, simple as that. And then, of course, whether or not fluid moves into or out of a capillary bed, it has to do with the pressure, the hydrostatic pressure, the pressure being exerted by the fluid inside of the vessel, inside of the artery or vein. So as the blood pressure forces fluid and solutes out of the capillary, so that pressure can actually force, as blood's coming into the capillaries, the blood pressure can actually force things out of the bloodstream, out of the arteries and veins and into the tissues, just the pressure of it. It's called the osmotic pressure. It's called the hydrostatic pressure. And then osmotic pressure is going to actually start to draw some of them things back in. So they'll leach back in by the pressure of their contents, what's in them, by those pressure gradients. So blood pressure is usually higher than osmotic pressure at the arterial end of the capillary beds. But at the other end, is the blood pressure is lower and the osmotic pressure is higher. And that's why the good stuff goes on and the bad stuff comes out. So, and that's what they're talking about here in this picture, or at least they're trying to show us that in this picture. The blood pressure here is much higher. So it's actually pushing fluids or pushing solutes into this tissue, even fluids into that tissue. But we get over here, the blood pressure is low and the solute concentration in the blood is low. So the bad stuff is going to start to flow back out. And it may even have some oxygen that goes back that way too. Just all depends on how saturated this is up here with waste products and oxygen and all that. All right? All right. And then, of course, as we're developing, there's just the heart is actually something very simple. It's called a tube heart, a heart tube. It's not much to it. It starts to become a 4-chambered organ in about 7 weeks, and it's beating. Most of the time, by the time someone finds out that they are actually pregnant and confirm it, by week 7, the heart is beating. Of course, there are some congenital issues that can happen, and they account for about half of all the infant deaths resulting from congenital problems. It's sad, but they're much more common than what we want to realize. A lot of people will talk about the high infant mortality rate that we have here in the United States, and they'll try to use that as a way of saying, hey, look how bad things are in the United States. Look how poorly they take care of their people. They'll use it as an argument for, all these hidden agendas that they may have. When the reality is The reason why we have a high infant mortality rate is because we take chances on infants that other countries don't. We invest and make an attempt to try to save children that other countries would just abort. They wouldn't even give them a chance. So it does raise our infant mortality rate. However, it gives many more of our little infants a chance. We try to save them. We try to give them a chance. But that explains why we have a little bit higher infant mortality rate. And there's some age-related problems. There's a whole host of these when we're talking about the cardiovascular system. Weakening veins and venous valves, varicose veins, atherosclerosis, the buildup of plaque inside the arteries, high blood pressure from loss of elasticity of vessels or any number of things, coronary artery disease from fatty calcified deposits in the vessels, that kind of stuff. So a lot of things happen. We can fix a lot of things by modifying our diet, stopping smoking, getting exercise, and doing all those kinds of things. They do actually help and prolong life, but it's not a guarantee. There are people out there that are very healthy that have heart problems, and there are people out there that are very unhealthy that don't have heart problems. It's not really fair, but I really think it all balances out, and it happens the way that it's supposed to be. All right, so that's chapter 11. I will see you guys back at chapter 12.